Hello, welcome back to the interesting world of microfossils. So far we have learned what microfossils are, how we study them with a stereozoom microscope, how we collect samples from the field, as well as how we collect samples from marine sediments and how we process the marine sediments for studying Foraminifera. Now we continue our lectures with how we separate the microfossils from the matrix of the hard rock. The separation of microfossils from the matrix involves a combination of mechanical and chemical disaggregation techniques. The exact technique will depend on the type of microfossil, that is, the chemical composition of the shell wall, and the nature and age of the rocks hosting the microfossil. The ground rule is the lesser the treatment necessary to free the fossils from the matrix, the better it is. As we mentioned, the technique used will depend on the types of microfossils. So, what are the different types of microfossils? The microfossils may be calcareous, like Foraminifera, the ones which we talked about before, ostracods, pteropods, calcareous algae, Capulets, Bryozoa. They may be siliceous microfossils like radiolaria, diatom, silicoflagellates, or they may be phosphatic ones like the conodonts, or they may be organic walled like spores and pollens. How do we process the rocks which contain calcareous microfossils? Well, there are several steps involved. The first step would be break the samples into fragments less than 1 centimeter in diameter with a rock splitter or hammer. Now let's divide the samples into 2 equal parts. We use one of it for further processing and the other part is preserved for reference. In the second step, we soaked the crushed samples. Now depending on the type of the matrix, different types of mediums are used. For soft calcareous shell or mudstone, we soak the samples in water. For moderately hard mudstone or marls, we boil the samples in water with a dispersing agent like sodium bicarbonate. For mudstones, we soak it in 50% hydrogen peroxide overnight and then we boil it for half an hour to 45 minutes. For shells, we dry, heat and plunge the samples into kerosene or gasoline. We keep on repeating these processes till the samples disintegrate. For the third step, we take the disaggregated material and then wash through a sieve with openings of 63 microns, which is an ASTM with 230 mesh size. The washed residue can then be dried on a hot plate or in an oven. We already talked about how we wash these samples before when we talked about processing the marine samples for foramininifers. To obtain calcareous microfossils from hard rocks like some argillaceous sandstone, shells and marls, we follow a bit different process. The smaller pieces of the samples are covered with a concentrated solution of sodium thiosulfate or mixed with an equal amount of crystals and then heated. When soaked with the solution, the sample is kept in a cold place where it remains until the salt crystallizes and forms a solid mass together with the rock fragments. The sample is then melted. This process is repeated several times until the rock is broken up by the growth of crystals in pores and fissures. The disintegrated rock sample is then washed until the salt is completely removed. For harder rocks like chert and siliceous sediments, complete removal of microfossils is difficult. So, the samples are best studied in thin sections or by immersing the thin chips of the material in alcohol or in immersion oil. Now, let's look at some of the important calcareous microfossils. The first one and the most important one to be mentioned is Foraminifera. Foraminiferas are unicellular marine microscopic animals with pseudopodia. Their tests are mostly calcareous. However, some are agglutinated and few are pseudo-chitinous. They range in size from 0.1 mm to 10 cm. There can be planktonic forams and benthonic forms also. The planktonic forms range from Jurassic to recent times, whereas we find the benthonic forams right from Cambrian age to recent times. Forams can be classified based on their character of shell. As we mentioned before, the shells can be agglutinated, calcareous or microgranular. The agglutinated test walls are built by cementing particles onto a layer of tectin. The cementing particles may be sand grains, tests of other microorganisms or calcite microgranules. The calcareous forams are actually of two types. They may have hyaline tests which have minute perforations on the test wall, or the tests may be porcelaneous. These porcelaneous tests have a smooth, shiny appearance. Microgranular tests are a link between the agglutinated and the calcareous tests of forams. The microgranular particles of calcite are cemented by calcareous cement and they characterize the wall type, giving it a sugary appearance. The forams are also classified based on their different types of apertures and the shape of their chambers. Apertures are the primary opening of the test to the outside environment. They vary in size and shape and is most often a function of the shape of the chamber on which they are located. In the pictures we can see different types of apertures. They may be the open end of a tube. They may be terminal radiate, terminal slit, umbilical, loop-shaped and various others. The external and internal structure of the chamber are important taxonomic features. The chamber forms may be spherical, globular, ovate, pyriform, tubular, angular, fistulose, etc. Now, forams can be single-chambered or unilocular or multi-chambered or multilocular. The chambers are added in a variety of patterns which is termed chamber arrangements. Here we can see the different ways chambers can be arranged. They range from cyclical to fistulose to planispiral involute, planispiral evolute, uniserial, biserial or triserial. There may be other forms of chamberal arrangement too. Forams may be found in planktonic habitat and also in benthonic habitat. However, there are only 40 species of planktic forams present. They have been successfully used in biostratigraphy. Their abundance helps understand the surface water conditions. Their isotope studies are used for local paleotemperature and paleosalinity reconstructions. Planktonic forams have been successfully used in biostratigraphy. Their abundance helps understand the surface water conditions. Their isotope studies are used for local paleotemperature and paleosalinity reconstructions. In contrast to the few species of planktic forams, there are over 4,000 species of benthic forams living at present, and more than 50,000 species has been described since the Cambrian times. The benthonic forams occupy every niche of the ocean, so their assemblages are studied to understand the paleoceanographic conditions. The stable isotope study of benthic foraminifera have been used for reconstructing the glacial and interglacial epochs. Now let's focus on another calcareous microfossils, the ostracods. In contrast to the forams which are found only in marine conditions, the ostracods are found in almost all aquatic conditions. These are small crustaceans, typically 0.5 to 5 mm in size. Their bodies are flattened from side to side and protected by a bivalve-like calcareous to chitinous carapace. The hinge of the two valves is in the upper or the dorsal region of the body. The ostracods grow by molting. The younger carapaces or the instars may be preserved in sediment with fully grown morphotypes. Sexual dimorphism is very common in ostracods. The two valves of the ostracods are joined by adductor muscles, and these leave attachment scars. The hinges between the valves range from single valve overlap to teeth and socket arrangements. The ornamentation is also highly variable. Classification of ostracods are based on these characteristics of the carapace, and they vary during the ontogeny and shows sexually related differences. The ostracods show both benthonic and planktonic modes of life, but planktonics have weakly calcified shells and are rarely fossilized. Only the benthics are preserved as fossils. They are found in all aquatic environments, freshwater, brackish water, and normal marine waters. The rapidly evolving ostracod lineages are extremely useful as markers in intra-regional stratigraphy. However, the warm water species cannot cross physical barriers, so they are of limited use for intercontinental or inter-regional stratigraphy. Ostracods are very sensitive to environmental change and respond rapidly to moderate variations in depth and salinity. So, they are excellent paleoecologic and paleogeographic tools. They range in age from Ordovician to recent times. Pteropods are another kind of calcareous microfossils. Pteropods are also known as sea butterflies. These are marine gastropods adapted to pelagic life. They have aragonetic shells that are very susceptible to dissolution. So, pteropod fossils are preserved only between depths of 300 to 700 meters. Scientists have seen the negative effects of ocean acidification on pteropods and are studying these organisms to better understand the problem. Pteropods range in age from Upper Cretaceous to recent times. Bryozoan colonies are another kind of calcareous microfossils that we might observe. The bryozoans are sessile benthic organisms chiefly living in marine environment. They live in colonies containing several microscopic individuals, but colonies may range in size from few millimeter to 10 centimeter in diameter. Majority of bryozoans have calcified skeletons that are easily preserved as fossils. They range in age from Ordovician to recent times. The bryozoan skeletons are often highly adapted to environmental conditions, thus making them invaluable for interpretation of ancient environments. The extinct Calcareous are another group of calcareous microfossils. Calcareous are pelagic protozoans possessing an axial symmetrical calcareous test. They resemble recent tintinnids. Tintinnids possess organic test as opposed to the calcareous test of the calcareous. Calcareous occur typically in very fine-grained limestones or marly limestones, which are very poor in benthic fossils but rich in microradiolarians and nanoplanktons. This clearly indicates that calcareous were also marine planktonic protozoans. They range in size from 30 to 150 microns, and they are found from Upper Jurassic to Upper Cretaceous times. The Calcareous limestones are very representative of Upper Jurassic and Lower Cretaceous pelagic species of the Tethyan province. Other than the types of calcareous microfossils described, we also find various calcareous algae as microfossils. These are calcified remains of aquatic, autotrophic, non-vascular plants. The marine varieties include cyanophytes or the blue-green algae, rhodophytes or the red algae, and chlorophytes or the green algae. The non-marine ones are charophytes. They range from Cambrian to recent times, and they are used in the interpretation of paleo-environments and for age determination of strata. Apart from calcareous microfossils, there are calcareous nanofossils, which are even tinier size, and the sample preparation technique for calcareous nanoplanktons are a bit different from the microfossils. For processing samples for calcareous nanoplanktons, distilled water is added to about 5 grams of crushed rock sample that has been passed through a 30 mesh sieve, and the mixture is thoroughly stirred. The sample is short centrifuged at 300 rpm for 15 seconds. The decantant is set aside. This process is repeated 3 to 8 times until the decantant is nearly clear. The decantant is short centrifuged again at 850 rpm for 30 seconds and then set aside for later examination. The residue contains particle size between 3 to 25 microns, rich in nanofossils. To prepare the strewn slides, the suspension containing nanoplankton is dispersed on a glass cover slip and dried. The cover slip is mounted on a glass slide with Canada balsa. Coccolithophore is the most important and major type of calcareous nanofossil that we find. When fine particles of marine sediments are observed under very high magnification of 1,000 times or more, a great diversity of tiny arrays of calcite crystallites are observed. These are the calcareous nanoplanktons. The coccolithophores are minute, dominantly marine, planktonic, autotrophic, unicellular, golden brown algae of extremely small size, even less than 25 microns. Their spherical cell wall, called the cocosphere, is covered with minute calcareous plates or coccoliths, which are preserved as nanofossils. These are mostly marine, but are also found in coastal and fresh waters. They range in age from Jurassic to recent times. Their rapid evolutionary diversification has made them extremely invaluable as biostratigraphic tools. In the deep sea drilling vessels, they help in rapid age determination of the deep sea cores obtained. Other than the coccolithophores, there are various other non-coccolithophore nanoliths. They are the Bryozoans. They range in age from the Cenozoic to the Mesozoic. The ceratolithids, these are horse-shaped and occurred only during late Cenozoic times. The discoasterids, these are disc or star-shaped astroliths ranging in age from Paleocene to Pliocene times. The lithospermationids, these are thick triangular tests with highly ornamented surface, and they occur during the Cenozoic time. Now let's come to another type of microfossils. In fact, let's first discuss how to prepare the samples to study them, the siliceous microfossils. Sample preparation techniques involve treatment of the samples with hydrogen peroxide to remove organic matter, then treatment with hydrochloric acid to remove carbonate fractions. The suspension is decanted and the residue is further treated with 25% HNO3 to remove the last traces of organic matter and inorganic salts. Distilled water is added to the residue and allowed to stand for some time. The solution is decanted and the residue is ready for preparation of the slides. To prepare the slides, the residue is diluted with distilled water, stirred well so that the microfossils are brought into suspension. Small amount of this suspension is spread on the glass slide and dried on a hot plate. They are fixed in glycerin for temporary mounting or with Canada balsam or hyrax for permanent mount. Silicoflagellates are a type of siliceous microfossils. They are related to coccoliths but with siliceous skeleton. These are marine planktonic organisms. They are single-celled, flagellate marine plankton with an internal skeleton constructed of opaline silica. Their skeleton may consist of thin rings, arcs, and spines. They may form complex ring of two rings or polygons. They may form complex rings of two rings or polygons joined by series of rods. They range in size from 10 to 100 microns. They appeared during the mid-Cretaceous times and diversified during the Tertiary, especially during the Miocene times. Their living representatives occur in all latitudes. Diatoms and Radiolarias are some other important siliceous microfossils. Coming to the diatoms, these are unicellular algae with siliceous external skeleton called the frustule. The frustules consist of two valves fitting together like minute boxes and lids. Their shapes and structures are extremely variable. They can be discoidal, elliptical, rhombic, triangular, rectangular, and so on. Diatoms can be solitary or colonial. They can be planktonic or benthonic, but are confined to the photic zone only in marine as well as fresh waters. Diatom frustules varies in size from 2 microns to 2 millimeters. They range in age from Jurassic to recent times. Diatomaceous earth is used in the industry as a filtering medium. Diatoms are used for interpretation of paleoecology or sedimentary rocks. Radiolarians are an important group of siliceous microfossils. These are a diverse group of pelagic marine protozoa, spherical or conical in shape, with radiating filiform pseudopodia. They construct elaborate biogenic siliceous or strontium sulfate skeleton. There are 3 major groups of Radiolaria, of which only the group Polycystina, that has a skeleton composed of amorphous opaline silica, is fossilized. There are 3 major groups of Radiolaria, of which only the group Polycystina is fossilized. Polycystina has skeleton composed of amorphous opaline silica. The polycystin Radiolaria are classified as spumillarians, which have spherical, oval or discoidal shapes, and nacellarians, which are bell-shaped, ring-shaped or bilobed. The Radiolarians live in open sea in all climatic zones. Their size varies from 50 to 2000 microns. They range in age from Upper Cambrian to recent times. Radiolarians are used for local correlation purposes. Now let's come to another interesting group of nanofossils. These are the phosphatic nanofossils, and prepare the samples in a different way to study these phosphatic nanofossils. We need a huge amount of sample to treat to study these phosphatic nanofossils because they are extremely small in size and are not so much abundant as the microfossils we spoke about earlier. 1 or 2 kg of rock samples is required for acid treatment. The crushed rock samples are immersed in 10% glacial acetic acid buffered with sodium acetate to a pH of 3.5. Alternatively, 10% formic acid buffered with calcium carbonate and tricalcium carbonate to a pH of less than 3.6 can also be used. The digestion may take a day to several days and require regular change of the acid. Conodonts are the phosphatic nanofossils, which we are interested to study. These are minute tooth-shaped isolated phosphatic hard parts of extinct group of primitive jawless vertebrates belonging to chordata. They have been found in rocks from Cambrian to Triassic age. Due to their very small size, they range in size from 64 to 200 microns, their abundance, widespread distribution, and their distinctive evolutionary transformations, conodonts are very valuable in biostratigraphy and are used as index fossils. The color of the conodonts may change from pale to black under the influence of temperature and time, and it is in proportion to the depth and the duration of burial. So, this conodont color alteration index is an indicator of the thermal maturity of the sediments, and it is used in basin analysis for hydrocarbon exploration. Now let's come to the organic walled microfossils. Let's first discuss their sample preparation techniques. The rocks are dissolved by a succession of chemical reagents. The crushed rock fragments are placed in a polypropylene beaker and are first reacted with hydrochloric acid and then with hydrofluoric acid to remove the carbonate and silicate fractions of the rocks. The residue is washed, centrifuged, and then reacted with Schulze solution, which is prepared by mixing 1 part saturated aqueous solution of KClO3 with 2 parts concentrated HNO3. The residue is washed and treated with 10% solution of KOH or NaOH to remove humic materials. The final residue consists of spores, pollens, microplankton, leaf and bark cuticles and other plant fragments. It is centrifuged, dried and mounted on slides with cover slips for examination under a light microscope. Other than spores and pollens, the important varieties of organic walled microfossils would be Dinoflagellates, Acrearchs, Tasmanids, etc. Dinoflagellates are unicellular biflagellate algae ranging in age from 5 micron to 2 millimeters. They have a varied habitat. Some are planktonic organisms in marine and freshwater environments, few are marine sand dwellers, and some are symbionts or parasites. Most dinoflagellates are autotrophic. They contain chromatophores and carry out photosynthesis. Some are heterotrophic and feed by ingesting their food. They have a simple life cycle which include a vegetative state and a encysted state or the resting cyst. Only the resting cyst stage is preserved as fossil. Not all species produce cysts. Therefore, the fossil records represents only a small portion of the whole group. True cysts have been known from Triassic times. Evolution was moderately rapid, which made them significant tool in biostratigraphy. Acritarchs, like fossil dinoflagellates, are microscopic fossil cysts, but it cannot be determined from what type of ancient plankton they were derived. The majority of acritarchs lack diagnostic features which might identify them as dinoflagellate cysts, but otherwise they are very similar to them. Their wall is believed to be composed of sporopollenin. They were first recorded in the Precambrian. They became the dominant phytoplankton group in Precambrian and Paleozoic communities, but they decreased in importance during the Mesozoic and Cenozoic times. Tasmanids are unicellular, relatively thick-walled organic fossils characterized by the presence of numerous fine radial canals which may pass partially or completely through the wall. They are predominantly spheroidal or more rarely ovoidal, reniform, biconvex or disc-shaped. However, the biconvex, disc-shaped or the reniform shapes were probably due to compression. The Thespanetids are cysts of planktonic organisms living in a restricted marine environment and sometimes forming marine peats. They are very common in exogenic black shells which has restricted circulation, suggesting that the environment of deposition was a death trap for them. Or alternatively, it may be that Tasmanites cysts lived in anaerobic toxic environments which restrict or inhibit the existence of other organisms. They range in size from less than 100 microns to 600 micron size. Their stratigraphic range are from Cambrian to Miocene times. Spores and pollens are the most important organic walled microfossils. The spores are produced by lower plants such as groups of algae, fungi, mosses and ferns, and some of the earliest preserved remains of plants. They are found Silurian onwards. Pollen grains are the male reproductive bodies or microgametophytes in seed plants. They originate on the anthers of flowering plants or angiosperms like oats, grass, roses, and in the microsporangia of gymnosperms like pines, firs and spruces. They are found from Devonian times. Due to chemical and mineralogical modifications during burial, their color may change progressively from pale yellow to orange to brown to black. Their color indicates the temperature regime of organic maturation in sedimentary basin. Pollens and spores of plants are allochthonous micropaleontologic group. They are products of continental vegetation transported to the place of deposition by wind and preserved in lakes or marine sediments. Other plant microfossils include seed coatings, seeds and wood fragments in thin section. Their study makes up this whole field of palynology. Identification of spore pollens and other organic nanofossils require high-quality compound binocular microscope equipped with oil immersion objective. They should range in magnification from 950 to 1500 times. Hope you enjoyed this interesting world of microfossils. Thank you for your time and patience. All the best for your upcoming lectures. Thank you.